Hello everyone, in this video we are going to discuss about gram-positive organism, that is Bacillus. So we know the gram-positive bacteria are divided into bacilli, cocci and branching filaments. So coming to the cocci, we have already discussed about all the gram-positive cocci. Most of the cocci are aerobic except for the peptostreptococci, which is anaerobic cocci. So these cocci are divided based on the catalase test. Catalase positive one are the staphylococcus and catalase negative ones are the streptococcus. Whereas the staphylococcus is again divided based on the coagulase test. Coagulase positive one is the staphylococcus aureus, whereas coagulase negative ones are the epidermidis and saprophyticus. We will divide this based on the neobisin sensitivity. Neobisin sensitive one is the epidermidis and resistant one is the saprophyticus. So coming to the catalase negative one, that is a streptococcus, it is divided based on the hemolysis on the blood agar. So it is divided into alpha, beta and gamma. Alpha means partial hemolysis. So these are divided based on the optician sensitivity. Sensitive one is the pneumonia and the resistant one in the viridans one. Beta hemolysis is a complete hemolysis. That is based on the Bacillus and sensitivity. These are divided. Sensitive one is the pythons and the resistant one is the Ehrlichia. Gamma hemolysis means no hemolysis. Gamma means no hemolysis. So here the organism is Enterococcus. So coming to the bacilli, so here the bacillus is divided into aerobic and anaerobic ones. Aerobic one is the Bacillus, Listeria and the Corynebacterium, and the anaerobic one is the Clostridium. Coming to the branching filaments, these are divided into aerobic and anaerobic. Aerobic one is the Nocardia and anaerobic one is the Actinomyces. In this class, we will discuss about the Bacillus in the gram-positive bacilli. So the Bacillus one is divided into two things, that is Bacillus cereus and the Bacillus anthracis. So Bacillus anthracis, firstly, it is a gram-positive rod. It is a spore-forming bacteria. It is aerobic and it is non-motile. It is encapsulated. It have a poly-D-glutamate capsule. It is non-hemolytic. And the toxins which are produced by this Bacillus anthracis are anthrax toxin, which have three factors, that is edema factor, lethal factor and the protective antigen. Coming to the edema factor, it will with the help of the adenylate cyclase, it will increase the cyclic AMP and causes edema. So that's why it is called as edema factor. Next is lethal factor. What it does is it will with the help of the zinc metalloprotease, it will cleave the MAPK, that is mitogen activated protein kinase, and causes cell death. And next is protective antigen. What it does is it binds to the host cells and it allows the entry of this both edema factor and the lethal factor. So this is the anthrax toxin. Next, the diseases caused by this are anthrax. That is, it can be cutaneous anthrax, like skin, affecting the skin, causing black eschar. Pulmonary anthrax, affecting the lungs, also called as Wool-Sorter's disease, and gastrointestinal anthrax, which is rare. The transmission is through contact with the animals, which causes this cutaneous anthrax, inhalation of the spores causing the pulmonary anthrax, and ingestion causing the gastrointestinal anthrax. The incubation period is in days, and the vaccine is there. Vaccine will be given for high-risk individuals like military personnel and lab workers, because they are more affected to this disease. So for high-risk individuals, we will give this vaccination. The treatment is it is penicillin sensitive, and the treatment can be ciprofloxacin or doxycycline with help with antitoxin can be given. Coming to the other species, that is the Bacillus cereus. So it is also a gram-positive rod and it is spore-forming bacteria. It is aerobic and it is motile, contrary to the anthracis, which is non-motile and it is non-capsulated, doesn't have any capsule. It is beta hemolytic, contrary to the anthracis, which is non-hemolytic. The toxins here are enterotoxins, that is, there are two toxins, that is heat labile and the heat stable. Heat labile means it is sensitive to the heat. The heat labile toxin is the diarrheal toxin. What it does is it increases the cyclic AMP and causes watery diarrhea. And the heat stable toxin is emetic toxin. Emetic means causing the vomitings. It acts like a staphylococcal enterotoxin and stimulates the vomiting center. So these are the two toxins, that is heat labile and the heat stable, and it also includes the hemolysis and phospholipases in the toxins. The diseases which are caused by this Bacillus cereus are food poisoning. It can be emetic and the diarrheal types. and also it causes opportunistic infections. The transmission is through ingestion of the reheated rice or any other contaminated food. Incubation period for the emetic type is 1 to 6 hours, and for the diarrheal type is 8 to 16 hours. There is no vaccination for this, and the treatment is supportive care. Antibiotics are not generally useful for this kind of food poisoning. So these are the important points in Bacillus, that is Bacillus anthracis and